A short break now
So it's wonderful to see all of you here. I miss seeing your faces every day, but we're just moved yesterday to one new city, so I'll start to be seeing you more. So glaucoma in African ancestry individuals is the topic of the POAG study. And it's interesting that glaucoma, which is present at a much earlier age in African ancestry individuals and is more rapidly progressive and 4 to 5 times more frequent and 15 times more likely to be visually disabling, has not been studied very much in this population. In fact, overall, all genetic studies, only 2% of genetic studies have been conducted in African ancestry individuals. The primary open-angle African American glaucoma genetics study, POAG, is a $17.85 million NIH-funded study that examines the genetics of primary open-angle glaucoma in these individuals. And so far, we've recruited 10,255 people with full phenotypes because all of them are from the Philadelphia region, they're 35 years of age or older, and they self-identify as Black. So the self-publication that Josh mentioned is called a multi-cohort genome-wide association study in African ancestry individuals. It reveals risk loci for primary open-angle glaucoma. And the commentary published alongside the article was making glaucoma genetic studies more diverse. And we were happy to see it in the press a bit because it made Black people aware that they should come in and be screened, because if you're 35 years of age when you get the disease and then you don't know because it affects your peripheral vision rather than your central vision to come into the doctor, it's really important that you be screened. And Marilyn Ritchie's picture is there because she was the bioinformatician who helped a lot in that portion of the genetics part of the study. So this is a graphical abstract. The study began with a discovery mega-analysis in cases and controls, and we used 3 African ancestry data sets, and then 46 genome-wide significant risk loci for glaucoma were identified, and then we did a sex-stratified analysis. And what you see to the right of that is a Manhattan plot, and the dotted lines represent significance levels. And as the Manhattan plot heads up, the significance levels rise, and that shows where the 46 risk loci are. And then we did post-GWAS analyses. We replicated in 4 cohorts. We compared across ancestries because most of genetic studies have been done in European people, although some in Asian. And we have traits for every single person who enrolls. So IOP, visual field, OCT, retinal nerve fiber layer thickness, visual field parameters. And we co-localized these traits with the genetics of the disease in this post-GWAS analysis, and then did fine mapping and looked at expression in eye tissues for these variants. And the outcome was that we were able to elucidate glaucoma mechanisms in African ancestry population. We found 3 likely causal variants, and we developed a polygenic risk score so we could see who was most at risk. And our polygenic risk score outperformed a European polygenic risk score, even though it was from a much larger cohort in African ancestry individuals, not completely surprisingly. So this is just detail about the study design. The discovery cohort, as you can see in the orange, was all with African ancestry individuals. It was POAG, adages, and GLAD. And then we replicated our results in GLAD2, All of Us, Penn Medicine Biobank, and the Million Veterans Programs, again, all self-identified Black. And then in the comparative analysis I mentioned, we used the global biobank from Europeans, East Asians, and all. And you can see that there's very few African ancestry individuals there. And this is a complex picture, but it really encompasses everything. If you look in the squares, you see the 22 autosomes that make up the human genome. And around the outside of the squares are orange, which are novel genes that we identified, those 46, and the blue are previously associated genes with glaucoma. And as you go deep to that, you see the Manhattan plots for every chromosome and where the significance level is. And the significance level for this investigation is the red dotted line. And then we did a sex stratified analysis in the green. And you can see that the larger the green circle, the more heterogeneity we identified. And centrally, we looked at the POAG GWAS, and then we used the Global Biobank Meta-Analysis Initiative for, as you go around, the Europeans and the East Asians, and then everyone. So this is in little more detail. Only 2 of the 46 significant loci were previously reported in other glaucoma studies. 21% of the 174 previously identified loci replicated at Bonferroni corrected significance. And if you look at some of the genes, you can see at the top of the first square that that ROC1P1 had a meta p-value of 1.67 times 10 to the negative 24th, whereas DB4P2 had a p-value of 1.12 times 10 to the negative 10th. So they were truly significant. And then, as I mentioned, we looked across ancestries, and we looked really around the world, and we saw substantial differences in the minor allele frequency for both replicating variants between African and European and East Asian populations. And what this does is highlight the influence of population-specific genetic factors in shaping allele frequencies. And it really underscores the need for interpretation carefully of genetic association studies which are conducted in different populations, because not everything replicates across populations. In fact, quite the opposite. Then we use, as I mentioned, the traits to co-localize with the lead variants. And one of them mapped to ARHGEF12, which has been associated in our study with baseline cup-to-disc ratio, but it had previously been associated with elevated intraocular pressure in individuals of European ancestry. So that looked like a great candidate gene. Then, as I said, we did cross-ancestry comparison, and novel variants from the mega-analysis have larger effect sizes in African ancestry individuals, not surprisingly. Previously reported variants, mainly from European individuals, have smaller effect sizes that point to the polygenic tail of POAG risk, as you can see at the right side of this slide. The lower genetic correlation among African and East Asian individuals was 59%, African and European individuals 73%, and European and East Asian individuals was 78%. And if you look at these graphs, you can see that in human trabecular meshwork cells, ARHGEF12 is significantly overexpressed. And there's significance also for, in the chart to the right, for the IPSI RGCs, and then human retina is significant for DBP-F4P2. So these we decided were the likely causal variants, and we analyzed quantitative expression profiles in human tissues using human trabecular meshwork cells. Shout out and thank you to my glaucoma friends who gave us these from glaucoma surgeries. And then we used induced pluripotent stem cells to form in our lab retinal ganglion cells, and then we used retinal cells. And we employed quantitative RT-PCR with and without H2O2 treatment to stress the cells. So here's what we we hypothesize. We propose that ARHGF12 functions downstream of TGF-beta and Rho-associated kinase signaling pathways to activate the ROCK1 via Rho-A. This decreases trabecular meshwork plasticity and aqueous humor outflow and increases RGC death and optic nerve atrophy. So what we found is ARHGEF12 is elevated, which elevates Rho-A. What is known is that that elevates ROCK1 from our study, which is the bottom level, and elevates the cup-to-dis ratio, resulting in increased RGC loss, increased optic atrophy, and increased cup-to-dis ratio. Now, the elevated IOP that was associated with this in Europeans was due to extracellular meshwork depositions, decreased aqueous humor outflow, and increased intraocular pressure. And then we developed a polygenic risk score for African ancestry individuals who are represented in blue. And you can see the performance of the polygenic risk score in African ancestry individuals was much significantly higher in overall compared to European individuals. So you can't really generalize using polygenic risk scores for Europeans to predict risk in African ancestry. And then when we looked at the people most at risk, those in the top 20% of their polygenic risk score, again, it was significantly better in predicting risk in the blue than the Europeans. So the next steps for the POEG study is quantitative trait association analysis with Marilyn Ritchie's group. Shefali Verma, who's gone on to become not only a postdoc, but now an assistant professor in pathology, is working on that. It's enhancing the predictive power of the polygenic risk score through deep learning models and meta-analysis techniques, which Marilyn is doing. Bingson Zhao, who's a bioinformatician at the Wharton School, is analyzing our exome-wide association analyses for binary and quantitative traits as well. And the whole exome data compared to the GWAS data, which gives us GWAS data we put on a customized chip. And so those are the common variants. And when you do the whole exome, you get the rare variants. So we'll be working on that next. And then we're going to evaluate significant GWAS loci in the context of three-dimensional genomics. So we know that as DNA coils and uncoils, that certainly different regulatory elements come in contact with different genes. And what's really exciting is that the work that has been done with the algorithm group, Marilyn Ritchie's whole lab, is being replicated with structural genomics. So we're using orthogonal platforms to confirm that. And this is just our new lab at One University City, which is across from Shea, if you look a little bit to the left. And the goal is to elucidate the genetics of diseases that overaffect African ancestry individuals. And the things we're doing now is epigenetic reprogramming with Bruce Cassander, who had a publication in Nature, and he expressed 3 out of 4 Yamanaka factors and got useful methylome and transcriptome and was able to reverse glaucoma in animal models. We got a asset grant to use AI for screening for glaucoma with Osbert Bastani and Rebecca Salo in my lab, who's also an engineer. We're exploring the eye-brain connection in Alzheimer's disease with Ali Hamadani and Bingson Zhao, and we've submitted a letter of intent to the Alzheimer's Disease Foundation. We're building a community advisory board to find out what diseases they would like us to use this large database in, and we sent in an NIH Build Up Trust grant for that. And we're sharing our data set across schools and disciplines right now with dermatology, looking at a kind of very socially disabling form of alopecia. So research is a team sport, and my gratitude to a great team that supports this work. Thank you. very much. Great question, Mara, and thank you for all your help with this study. Mara is, along with Edie, was the face of this study, and I really appreciate all you've done for us. So yes, absolutely. We recognize that there are many factors that likely contribute to many neurodegenerations. And so we use census data to just get to publishing a paper and a technique called geocoding to look at that census data and see what demographics. We had a survey, of course, when people enrolled, as you know, but the survey is not as robust or rigorous as the geocoding method to see what the demographic challenges are for this population that could contribute to a neurodegeneration.
Dwight.
So, Joan, can you assess whether the genetic architecture of, sorry, can you assess whether the genetic architecture of African American glaucoma is different than Europeans?
It's quite different, and it really is quite different because when I did that Venn diagram, when you see all the genes that have been associated with glaucoma in Europeans, the overlap is really only a very small number of genes that were discovered in our study. So we think that ancestry plays a huge part in the genetics of disease. And so that may be particularly true in neurodegeneration. So the big goal is to personalize medicine the way we would for cancer. So you wouldn't treat breast cancer without first getting a full genetic diagnosis and then a personalized plan based on that diagnosis. And that's where it has to get with neurodegeneration, I believe.